And welcome all you future microbiologists. Now, I want you to think of this not as just some boring syllabus breakdown. No. This is your mission brief. We're about to map out your entire first-year expedition into the incredible, invisible world of microorganisms. This is your guide to navigating the core course, General Microbiology. Okay, so let's dive right in. This document right here, this is basically our map. It officially lays out the General Microbiology core course for your first year of your BSc, and it really sets the stage for everything you're about to discover. Every single unit, every practical experiment, it's all in here. So here's our plan of attack for today. We're going to start with the big picture objectives, then we'll chart out the whole theoretical journey. We'll meet the pioneers who started it all, zoom right into a bacterial cell, get you geared up for the lab, and finally, we'll look at how your success on this mission is actually measured. Let's get to it. All right, first things first. Any good mission needs to have really clear objectives. So what exactly are you expected to know and be able to do by the time you finish this course? Let's take a look at the official briefing. Take a look. This slide lays out the core skills you're going to have by the end of your mission. And this isn't just about memorizing a bunch of facts. It's about being able to tell the whole story of microbiology, from its ancient roots in India all the way down to the tiny, intricate machinery inside a single cell. Okay, now let's zoom out and get that 30,000-foot view. The theory part of your course is like the main expedition, where we map out the entire microbial landscape before we even think about getting our hands dirty in the lab. Your whole journey is broken down into 5 key units, and it's designed to build your knowledge in a really logical way. You'll start with the history of the field, then move into how we classify all these different life forms, and then you'll dive deep into the structure and growth of the bacteria themselves. Our first major stop is a journey back in time. We're going to explore the incredible history of microbiology, going all the way back to its ancient roots, and, of course, celebrating the pioneers who gave us the tools to finally see this invisible world. You know, it's the most basic question, isn't it? And this section is going to answer exactly that by looking at the trailblazers. You'll learn all about the key experiments and those eureka moments that really established microbiology as a science. We get to travel through this incredible timeline of discovery. We'll start in ancient India, exploring concepts from Vedic texts and Ayurveda. Then we jump to Antony von Leeuwenhoek, the draper who was the first to see what he called animalcules. And of course, we'll cover the absolute titans of the field, Louis Pasteur and Robert Koch, whose work completely revolutionized medicine and our entire understanding of disease. You know, discovering this invisible world, that was just the first step. The next massive challenge for scientists was, okay, how do we organize all of this? And that's where classification comes in. It's how we create order out of what seemed like chaos. So you'll get to explore how we've tried to organize all this diversity. You'll start with the classic system most of us know, Whittaker's 5 kingdoms. But then you'll see how genetic analysis led to Carl Woos's revolutionary 3-domain system, archaea, bacteria, and eukarya, which is really the foundation of how we see life today. And the journey just keeps building from here. We're zooming in. We went from the entire history of the field to how we classify everything. And now we're going even deeper. We're going right inside a single bacterial cell. This unit is basically a deep dive into bacterial architecture. You're going to study all their different shapes and arrangements, and you'll explore all the essential parts, from the cell wall that gives them protection to the flagella that let them move around. It's all about understanding how the machine works. But listen, it's not just about what a bacterium is, it's about what it does. And this is where you'll master the bacterial growth curve. You'll understand how a population just explodes with growth and then declines, all through these 4 distinct phases. Trust me, this single concept is fundamental to, well, everything from food spoilage to fighting disease. All right, let's step out of the lecture hall for a minute and head into the lab. Theoretical knowledge is absolutely vital, but your mission just isn't complete without some crucial hands-on field training. This is where all those concepts become real. See, your course is perfectly balanced. You've got the theory paper that we've been talking about, and then you have the practical paper. Each part is designed to reinforce the other, making sure you can not only explain these concepts, but actually apply them using real scientific techniques. And this is where the science gets really tangible and honestly super cool. You won't just be working with some abstract cultures. You'll be isolating lactobacillus from curd you eat every day, finding yeast on fruit, and actually testing how something like pH affects bacterial survival. This is microbiology you can see, touch, and, well, in the case of curd, even eat. Okay, and finally, let's talk about how you're graded. How is your progress on this mission going to be tracked? Let's just demystify the whole assessment process so you know exactly how to show that you've mastered this material. It's a really straightforward breakdown. The biggest chunk of your grade, 70%, comes from that final university exam. The other 30% is based on your continuous evaluation all through the semester. So what that means is your day-to-day work and your final performance both really matter. So that 30% for internal assessment, that's going to come from things like class tests and presentations. And the final university exam will test your knowledge with all kinds of questions, from quick recall answers to those more in-depth, long-form explanations. This whole structure is set up to reward both your consistent effort and your deep understanding. And it all comes full circle. Remember that mission objective from the very beginning? Every single lecture, every lab, and every exam question is designed to build these core skills. You're not just learning stuff, you're becoming a capable scientific explorer. So, there you have it. The map is laid out, the mission objectives are crystal clear, and the tools are ready for you. You've got a comprehensive plan to tackle every single part of your first-year syllabus. So, the only question left is, are you ready to begin exploring the unseen world?